Hey there, everyone. We're going to go ahead and let folks kind of come into the Zoom room as we get closer to our start time. In the meantime, we'd love to hear from you. Go ahead and let us know in that little chat box at the bottom of the Zoom window whether or not you've been to a Vernier webinar before. I hope everyone's, what is today, Wednesday? I hope everyone's Wednesday is going great. Very nice. Thanks for coming back, Jack. Very cool. Well, I'll kind of hop into my spiel really quickly. Thank you all so much for joining us for student-led explorations into respiration rate and spirometry. I'll pass it off to John here in just a moment, but I just wanted to cover a few housekeeping items first. First and foremost, the recording of this webinar will be live on our website and emailed to you as soon as it's ready. This typically takes about a week or a little bit longer, but you will get that recording and all the resources to your inbox, so keep an eye out for that. We would love to answer all of your questions today. So just like you've been putting chats in, go ahead and put your questions in that chat. I'm going to be keeping an eye on that, and I'll be answering what I can. And if I can't answer, I'll go ahead and stop John and we'll ask the question together. So if we don't happen to answer your question, we'll follow up separately and make sure you get an answer to that. With that being said, I'm going to go ahead and start a poll. It should pop up in your window. And as you're answering that, John will go ahead and start chatting with you all. So thank you again for coming, and I hope you enjoy your webinar.
Thanks, Dylan. All right, so I think a little poll is going to pop up. It would be great if you would answer that poll. The reason why it's great for you to answer that poll is it really helps me focus on where we're going to go next. So like, are you here mostly because you're interested in the sensors that we're going to be talking about, or is it mostly that you're interested in different Vernier solutions or software? That really helps. If I know, like, because if you're here to see sensors mostly and you want to know about the sensors, I focus more on that. If you're interested in all the different software solutions, I can focus on that later as well, or build that in. And then it's also really good to know whether or not most of you are high school teachers, college teachers, or other, because then I can focus on the different programs and different ways to use the sensors in those different spaces and places. So please fill out that pre-webinar panel, and then Dylan will let me know what we got. And then once we have that, great. Then this is what we've been finding when we do these type of polls, and it's interesting that it's been very consistent. Is most of you are high school, but we do have plenty of college, and we do have some of other, and that's great. We can cover all of that, but most of you are here and are interested in the sensors that are involved in this. So that's what I'm going to be focusing on, which is what we plan to do. So that's great. So let me close a little pre-webinar poll, and then let me take a look at the chat here. Oh, that's fine. I mean, a lot of what we'll do when I talk about the sensors themselves is the software that is used with the sensors. A lot of the stuff that I'm going to be talking to you about. So let me just back up and we'll start, and then we'll go a little bit farther here. So the topic of the day is what we call student-led explorations into respiration rate and spirometry with running tacked in on the end. And I'll explain to you why running is important and why that's actually a cool thing that we can take a look at and a little bit surprising. So you're going to meet me. Today's agenda was you're going to meet me. My name is John Melville. I'll introduce myself. I'm going to give you an introduction into the Go Direct Spirometer and the Go Direct Respiration Belt. So they're both Go Direct products, the Go Direct Spirometer and the Go Direct Respiration Belt. We do have older products, a respiration belt and a spirometer. It doesn't mean that you couldn't use both of those products for the experiments in the ways that I'm talking about it with graphical analysis. But I just want to let you know that's what we're going to be focusing on, is the Go Direct ones. But if you have the older ones, stick around, stay here, let me know, throw it in chat, and I can make sure that we discuss how you would use your older probes as well. I am going to talk briefly about options for remote education for remote education for in the classroom, synchronous and asynchronous environments. But mostly it's going to be a hands-on demonstration. Don't forget to put your questions in chat, and Dylan has already told you how to view the webinar recording after today. All right, so my name is John Melville. I have a bachelor's degree in biology and psychology. I went to Sonoma State University. I also have a PhD in zoology from Oregon State University. I did postdoctoral research in neuroscience at the University of Oregon. And then I was an assistant professor of biology at Wartburg College. And I focused on teaching majors and non-majors courses in biology, but I've taught a lot of stuff. But my big focus was on active learning with an emphasis on data collection and inquiry. My favorite thing being getting students to do their own experiments and design experiments to test with equipment. And that was one of the reasons why I loved working with Vernier products. All right. So for chat, don't ask all your questions in chat. Don't worry, someone will be here to answer them as we go. If there's stuff that I don't answer, we'll talk about it at the end. If you want to play with graphical analysis right now while we're talking, or if you want to, you can go to our website. You can go to downloads and you can get graphical analysis for free. And you can turn it into Graphical Analysis Pro by using this. This doesn't say this says Gangsta 223. It's GA NSTA 23. So what we're giving you is the NSTA code. When we were at NSTA, we had a code that we all use that'll be good for like another 2 weeks or so. And you don't need any sensors. You can install that on your devices, and I'll be able to send data to you if you're interested or want to. Let's see, do we have any questions? Thank you, Dylan. And if you have any problems getting to graphical analysis, I can show you how to do that. You just want to go to our website, go to vernier.com. You tap up here on the screen at the top right corner where it says download support, login, register, tap on downloads. You should see Vernier Graphical Analysis, and you can just tap on downloads. And it will auto detect which device you have, like whether you have a Windows device or Mac device, and you can download it. We also have it for Chrome. If you have an Android device or an iOS device or iPad store, these will take you to the proper store that you need to get it. All right, so let's go back to the demo here. Any questions? All right. So once you launch graphical analysis, you just need to put that GA NSTA 23 down here. So install graphical analysis, then launch it, and then put it in there. And I can actually even show you that here. I can go File, New Experiment. You can see it's looking for the activation license code here. I'm going to type in GA NSTA 23, submit. And then you'll see it's going to say, hey, that's the Vernier NSTA 23, and the subscription ends on February 28th. So you'll have it almost for not quite a month, but several weeks. All right. So let's go back to the presentation here. So what I wanted to talk to you about was two sensors that I think are really great for experiments. Look in human physiology. One is our Go Direct Respiration Belt, and the other one is the Go Direct Spirometer. The Go Direct Spirometer is a spirometer, so it's great for looking at lung volume parameters, tidal volume, etc. But it actually has some other cool features. And then there's the Go Direct Respiration Belt, which also is great for lots of different experiments. It's a little bit simpler to use than the spirometer, and we can talk about that. Someone previously, before I did this webinar, asked about price. So what I did is I have the website here, and you can see that the Go Direct Spirometer in the United States costs $219, and the Go Direct Respiration Belt costs around $100. So there also is a large difference in price, but the Respiration Belt does a lot of great stuff. You can do a lot of great experiments with it. The Go Direct Spirometer, if you're really interested in spirometry, that's what you want to do. This is the device that you want to use. I also wanted to just briefly say that there are 2 books that I highly recommend. This is our Human Physiology Experiments book called Volume 1, and then our Human Physiology Experiments Volume 2 book. Human Physiology Experiments Volume 1 has some great experiments in it for the respiration belt. Just take you to those here. If I scroll down and tap on Table of Contents, we can see which sensors are actually used. There's a really good experiment looking at respiration and ventilation, and then another one looking at ventilation and heart rate. Those are 2 really great experiments, simple to do. I can show you how those work today in our demonstration. And then also, the Human Physiology book Experiments 2 has some really good experiments in it as well. This book is the one that has the spirometer experiments in it. Let me take a look here at chat. I'll link to Human Physiology Experiments. Great. So let's take a look at the Volume 2 book. This book is what we call our, it's Volume 2. It's more advanced. This is what we would recommend. So like if you're at a college, what we would recommend that you do is get both books. If you're at a high school, what we would recommend is definitely get Human Physiology Experiments Volume 1. But if you are going to be using a spirometer and blood pressure sensor, definitely get the second book as well. They will work for in both cases, college or high school. The table of contents for book 2 includes blood pressure, some more advanced EKG labs, diving reflex, and then the spirometry labs, which we have lung volumes and capacities, spirometry and body position, respiratory responses to physiological challenges, an analysis of lung function. There's a way to simulate asthma. And then really interestingly, this analysis of barefoot running, which I'll talk to you about. And that uses the Go Direct Respiration Belt. There also are some other interesting activities like biofeedback and the diving reflex that use the EKG sensor and the Go Direct Respiration Belt. So don't forget, you can use these sensors with other devices at the same time too. All right. So let's just stop and back up. And what I'm going to do is I'm going to go through some of the basics of the Go Direct Spirometer and how you can use it to study what we would call classic spirometry, and then also use it to look at respiration rate and do some more long-term experiments. So everybody that I know that wants a spirometer, they usually want to do this lab down here, this graph that you can see in the bottom left. This is looking at flow rate and then tidal volumes and then vital capacity. That is like, I would say 90% of what people want to do is this lab. That lab is in our Human Physiology Experiments Volume 2 book, and that uses the Go Direct Spirometer. But there are lots of other experiments that you can do with the spirometer as well. So there's a good question. So the Go Direct Respiration Belt has experiments for it that are in Volume 1. So if you don't have a lot of money, just want to get started, you can not get the spirometer and get the Go Direct Respiration Belt, and you can do lots of great activities with the Go Direct Respiration Belt. Volume 2, Human Physiology Experiments Volume 2, is going to have more advanced experiments in it. It's going to have the spirometer, it's going to have the blood pressure sensor, and then also some other labs in it as well. If there are specific labs that you are interested in that you want to try, like 1 or 2, and you already have these products, just get in touch and I'll make sure that I can get you a copy of 1 or 2 labs. Like not the whole book, but I can easily, like if you're like, oh, I want to try this one lab, just let me know. You can email biology@vernier.com and ask, and I'll go find it for you and make sure that you have, a copy to play around with to see if that's what you want to do. Don't worry, we're gonna get to all of that stuff with the spirometer. So let's just talk a little bit about the spirometer. But before I get to that, I just want to say Vernier's, this should be Vernier's Science Education. We've changed our name to Vernier Science Education. It's for teachers and by teachers. I'm going to show you Vernier Graphical Analysis Pro. We're going to do a live demo. Don't worry, no sensors are required. Now the Go Direct Spirometer, it's great for comparing and analyzing respiratory patterns. It's great for measuring tidal volume and other lung volume parameters and analyzing lung function. But it also has some really cool special channels. It has a special channel to control for baseline drift, which is a classic thing that you're going to need to do. And then it has another special channel for longer experiments that I can show you. So if you want to actually do student-led experiments, both of these extra channels are going to be great things to have access to. All right. So what someone asked, let's just talk briefly about what a spirometer is and how it works. So what I have right here is I have a little camera set up. Here's the Go Direct Spirometer. You can see that it says inlet right here. It also says no water, so don't get it wet. You're going to need to use a bacterial filter. This is the bacterial filter. The bacterial filter goes on one side of the spirometer. Fits in really nice. It creates a nice airtight, not quite airtight, but a nice seal right there. You're also going to want to use a mouthpiece. We have these disposable mouthpieces that it comes with. There is also a thermoplastic mouthpiece that I like from a company called AM Systems that is not disposable. That works really well. It's a little bit more expensive, but if you're interested in that, once again, just email biology@vernier.com and say, hey, John, where can I get that thermoplastic mouthpiece? And I'll find out, and I'll send you a little link. You may want to tape this so that there isn't a lot of leaking when you use it, but it should work fine if you just seal it. And so then there's the spirometer right there, all ready to go. Now I'm going to show you this. Hopefully you can see a picture of me in the video. Here's me with the spirometer, right? Now, almost everything that I'm going to be showing you here about how to use the spirometer, you can find on our YouTube. You can find other videos of. You can see me when I was 10 years younger using a spirometer. All of these key things that I'm going to be talking about on how to use a spirometer, it doesn't matter if it's our old spirometer or our new spirometer, they still are relevant. The biggest thing is you want to hold it in the position that it's going to be used. So if I'm going to be blowing into it like that, I want to be putting my hands up and holding it steady. I don't want to be rocking back and forth. You don't want to rock back and forth with the spirometer. So that's crucial. The other thing is people ask, oh, hey, do I need this bacterial filter? Yes, it's one per subject. There's no getting around that. This bacterial filter also has a viral filtration efficiency. It's equivalent to an N99 mask. So it's really good at filtering out viruses and stuff like that. So it is safe to use. But remember, the bacterial filter, or this filter, which also filters out viruses, is actually there to protect the student from the spirometer and the spirometer from the student. So what we say is it's one per subject. Now, that doesn't mean you have to throw this away after a subject uses it. What I would recommend doing is writing the subject's name on it. So what I do is I write my name. Like if you look over here, I write my name on the bacterial filter, and then I wind up reusing it. All right, questions here. Do you need a mouthpiece? That's a good question. I've used the spirometer without a mouthpiece. You probably can get away without using a mouthpiece. It's just nice to have a mouthpiece, you know. But I demo them without a mouthpiece all the time. I would definitely recommend using a mouthpiece. The other really important thing about using it, and I'm going to turn sideways here so you can see me, is you don't want to blow on the spirometer like it's a trumpet. So you don't want to put your lips up to it and then blow. You want it, the opening of the spirometer actually goes in the mouth. So that's how you use the spirometer. And then the most important thing is to just try to breathe normally. Let me launch graphical analysis and we can take a look at it here. All right, so here's graphical analysis. Let me change how we view it here. About, actually, app preferences. Make it a little bit bigger. It's a little bit easier to see for you all. Now the Go Direct Spirometer, all Go Direct devices have a USB port and they have Bluetooth. The simplest way to connect them to your device though is through the USB port. So I'm just going to connect via USB. There we go. And then I'm going to minimize that. And what you can see here, let me just maximize this a little bit, is graphical analysis already sees that I'm connected to the spirometer. And it's already set up and said, hey, it knows what sampling time to use. It's already turned on different channels for flow rate and volume. So it's actually ready to go to do an experiment. One of the things to be aware of, though, like I talked about, is if you tap down here in the bottom right corner where it says sensor setup, there are different sensor channels, and we could talk about those later. So the default channels are going to be flow rate and volume. Another thing that I'm just going to say that would be useful to turn on is the adjusted volume channel. We'll talk about that later, but I'm just going to hit done. And then I'll do the classic standard breathing experiment for you. Let me see, I've got a chat question. Someone asked, is there a risk of data dropping if you do Bluetooth? There is, but it really just depends on the Bluetooth radio. I've used these devices over Bluetooth. I could use them over Bluetooth now. It works fine over Bluetooth. It's just, if you just want the simplest way to get started, use it over USB. And I'll show you how to connect it over Bluetooth next. So I'm just going to be breathing into this a little bit. We're going to be taking a look at some data and then we'll analyze it. All right. Oh. So let's just take a look at some of this data really quick. So this is the flow rate that I've shown you up here. And that's not really crucial for our analysis. I mean, I could analyze the volume. I could analyze the integral of these flow rates, but I'm just going to tap up here and I'm just going to say, hey, let's just show one graph. And then let's just show the volume. Great. So here's the volume, and I can like take a look at right there. Well, that's about 1.14 liters. That's about 1.24 liters. That's about 1.31 liters. And then some of you might be like, oh, look, it like came back down again. And it says that's 0.83 liters and that's, 0.85. Now, there is an easy way to do what we call a baseline adjustment here, and that's just to go from peak to trough. So if I were to go from peak to trough, and then under graph options, tap on view statistics, it's going to show me the delta Y, which you can see is about 1 liter. And for me, trying to breathe normally and having a foreign object in my mouth, most people would say, oh, it should be about half a liter. You're always going to see tidal volumes that are going to be larger than normal just because of the fact that your students have a foreign object in their mouth. Once again, I can take a look here at peak to trough, and I can view statistics. That's about 1.153. If I wanted to look at vital capacity, that's from the greatest inhalation to my greatest exhalation. And once again, I can just do view statistics, and that's a 2.839. Not very good. Not very good for me. Should be closer to like 4, but I'm out of shape. So probably not pushing enough air there. Now, there is a way, like I told you, I had this other channel turned on called adjusted volume. And what that does is it automatically adjusts. So you can see here, like from peak to trough, right? After that comes back down, it's like, oh, I'm at a baseline. I'm going to re-zero myself. So it re-zeros itself every single time. So in this case, I can just pop along here and see like, yeah, that's 1.14, which is close to what I measured previously. 1.12, right? Close to what I measured previously. A little bit higher, 1.23. And then you can see there's 2.3 and then about a negative half liter there. But what's nice is after my maximal inhalation and exhalation, it's auto correcting afterwards. So instead of having to take the difference, I can just come along here and measure directly. Now that's about 1.4, 1.4, and 1.5. So that's what the adjusted volume does. There's volume and adjusted volume. The questions on that so far? On just how to use the spirometer basics. What's nice is if I wanted to do a long-term experiment, I could turn on these other channels. And if I turned on cycle volume and respiration rate, let's actually just do that for a minute. So there's a question here really quick. If you have the old probes, no, if you have the old probes, that channel that I showed you right there is special. That's a really good reason to get the new probes, is that the Go Direct sensors actually have that auto correcting ability. The other sensors do not. So hold on here. Let me turn off that and turn off that and that. So here's cycle volume and respiration rate. That doesn't mean they still won't work if you have old probes. You still can do that trick that I just showed you. The biggest trick, if you have the old sensors, the old spirometer, is zero it before you use it, zero the flow rate, and then you'll have a lot less drift. But same thing, just measure from peak to trough. If you measure from peak to trough and get the stats there, you're going to get great data and it'll be fine. That's what we used to tell everybody all the time. And the older spirometers work great, and they have a flow head that can be removed and they'll last a really long time. So it's not like you need to get rid of them, and your old spirometers will work fine in graphical analysis. There just isn't that automated correction factor. Now, what's neat about these channels is if I turn on cycle volume and respiration rate, what it'll do is it'll calculate the respiration rate and it will just find the cycle volume and plot it as 1 point. So let me just show you what that would look like. So I'm going to hit collect. So if you wanted to do a really long-term experiment, and you didn't need to collect a ton of data, what it would wind up doing is for each breath in and out, it's going to find the cycle volume, the tidal volume for you. We call that cycle volume, 2.3, and it will plot that. And then in addition, what it will do is it will find each breath, and then over a period of like 10 seconds, it will then say, hey, this should be the respiration rate, and it will plot that too. So if you wanted to have students do their own experiment or create an experiment where they looked at changes in tidal volume and changes in respiration rate, the spirometer could be great for that as well. You could record for a lot longer, and you don't need to use those other channels. So other questions? If there are no questions, I'm going to disconnect the spirometer and we'll go back to the presentation really quick. Oh, just to remind you, if you're doing that classic experiment, I just wanted to let you know that you can get the tidal volume, you can calculate tidal volume directly, you can calculate vital capacity directly, which also means you can calculate inspiratory reserve directly. You can measure that. So one of the great things about our new lab book is it has you do that. And then you just get ERV, I believe. All right, so I just showed you that, how to actually collect real data. Yeah, so we can actually measure IRV directly. So you get a lot better data in our new lab book, in where what you do is you just calculate ERV, where ERV, expiratory reserve volume, is going to be vital capacity minus IRV and tidal volume. So we've modified the new Human Physiology Experiments Volume 2 so that you do that. It used to be that we had you sort of estimate IRV and ERV, and it just isn't as accurate as calculating VC and tidal volume and the others, and then calculating it. Now, once again, the spirometer labs would also work in our new Human Physiology book, would work with your old probes as well. All right. So if nobody has any further questions on spirometry and how it would be used, feel free to ask them now in chat if you want to. I'm going to switch over here, and we're going to talk about the respiration belt. And here's the respiration belt. Show you what the respiration belt looks like. It's a really quite simple device. It's really just a force sensor that has some straps on it. And you can see right here, the force sensor or load cell is in here. And what happens is every time you pull on it, you know, there's a force that's applied. And so you'll wind up looking at what we call respiration effort, and then it will also calculate respiration rate. All right, someone had a question. What question did they have? There is only, so if you have the older book, if you basically have the older Human Physiology with Vernier book, that's different. You would need to get the new book. You don't just get free copies of the new books, because the two books are very different. But if all that you need is just one lab, right? All that you need is just one or two labs. Just like I said, email biology@vernier.com. I just did that for someone today that had their old blood pressure sensors, and they wanted to see how they would work in graphical analysis. All of the labs can be modified in Google Docs or as a Word doc, and I can show you how to just simply modify them to make them work for you if you have our old probes. So if you need just one or two, just get in touch. Or if there's like a few that you want to test out, please just get in touch. Email biology@vernier.com. Just ask for Melville. Say that you came to this webinar and just tell me what labs you're interested in, and we could talk about them. All right, so the Go Direct Respiration Belt, it has a USB port on it. It also has some other cool features. Now I'm going to show you how to connect to it wirelessly. So I'm going to tap on it here. And you can see it's flashing next to the Bluetooth symbol right there. That means it's broadcasting and you can connect to it. Another thing that's neat is, let's see. Oh, there we go. When I pull on it, you see that little green, you see the check mark down in the bottom left corner? There's a little green LED that goes on. That means there's adequate force applied. So that's a good thing to look at, is when you strap this on yourself, you want to make sure that the students see that little light go on. That means there's adequate force that's been applied to the individual. All right. And let's go back a little bit. And what we should wind up seeing, this is an example experiment down here below. And what you could see is we should see a respiration effort. So as I breathe in and out, force is going to go up and down with each breath in and breath out. If I breathe faster, the respiration effort, these little spikes get faster. This is a breath hold. And then what's really interesting is after the breath hold, look at how much more respiration effort there is, right? And that makes perfect sense if we're talking about homeostasis. But then it will also calculate respiration rate. you can see the respiration rate gets faster as I'm breathing faster. And then when I take a deep breath and stop breathing, you see it falls to 0. And then the respiration rate is lower than it was previously. All right, question. Thanks, Judy. Yeah, if you have an account with Vernier, you can just log in and it'll show you what books you have and you can get the latest downloads. They should be there. You should be able to see what books your school actually has. It's incredibly useful. And if you want to know how to log into your account, just let me know. I can show you. All right, so the Go Direct Respiration Belt is awesome because you can use it to look at respiration rate changes after exercise or breath holding. And how respiration effort, that's the force exerted on the chest during respiration, changes also after exercise or breath holding. But it also can be used to measure steps and step rate, which we'll get to at the end. Once again, these experiments are found in these 2 books. These are the 2 books that I would recommend getting. And what I'm going to do first is I'm just going to show you how to collect data in real time. And then I'm also going to show you some videos that we have that are actually in graphical analysis of some of these experiments. So one of the cool things about Graphical Analysis Pro is you can stick video in it, you can data share with it. So we have some of these experiments actually in GA Pro. They're written for biology with Vernier, not human physiology, the human physiology experiments, but they're very close to the same. All right, so let's just go back to the live demo. I'm going to do file new experiment. All right. So I'm going to put the respiration belt on myself really quick. There. I know that the light is actually now green. I'm going to put it on pretty tight because I'm wearing a bunch of stuff. There we go. And then I'm going to connect over Bluetooth, and you can see that there is only one respiration belt. GDXRB is the order code in the room. And I'm going to tap on connect. And what you should be able to see here is it's got some other channels there, steps and step rate, but it's connected. We have a question here really quick. Yeah, it depends. Let's see here. Could you talk about placement of the belt? Yeah, I could talk about that. Judy, I don't know if I'm going to talk about data share. We can at the end if we have time. The green light doesn't have to be on the whole time, but it is useful if it is. What I would do is just tighten it, have the students like breathe in, hold a breath, or you can have them exhale and tighten it. Right now I'm breathing in and breathing out and the green LED is on the whole time. It doesn't have to be on the whole time, but it's going to be useful. So let me just show you. I'm going to take some just normal breaths. I'll try to breathe faster. Now I'll hold my breath. And believe it or not, that's like the first part of the classic experiment that we have you do. So let me tap here where it says zoom to all data. Do the same thing here. And then what students can do is they can go through and they can look at, well, what was the peak respiration effort? It's going to be very similar, right? They could just look at, well, what's the amplitude? Go from peak to trough. View statistics. And you could see it's about 15 newtons, right? And that if as I breathe faster, I'm not breathing as deeply. So in this case, you can see the amplitude decreases, but there's more of these peaks. And you can see the respiration rate increases. Once I take a big deep breath, I'm not breathing at all. It drops straight down to 0 because it sees this big change, and it's like, well, I don't see anything happening. So then it goes down to 0. What's really interesting though, as you can see, is that I have all these recovery breaths. So the first breath that I take after a breath hold is bigger than my normal breaths, which makes sense. I'm recovering after holding my breath. But what's interesting, or I think is really cool, because this is very reflexive, is you'll have a recovery breath, another recovery breath that's probably bigger, and then it will begin, your breaths will begin to look just like they did previously. And you can do all sorts of experiments. You could look at, okay, well, what happens if I'm rebreathing? That's another experiment that we have. This little sensor works great for lots of things. Questions about how to use the respiration belt? And I've done all this on Bluetooth just to show you how to use it on Bluetooth, but you could also use it over USB. And then don't forget, you could use this respiration belt with an EKG sensor or something else to do other experiments. So you can use them all together. All right. Really, really useful little sensor, really simple to use. A lot of people ask me like, well, can I use it? Well, we'll get to that. The next thing I'm going to talk about here is other ways that I can use it. So just to show you again, there is another experiment here that's very similar. I just want to show you that there are experiments that live in GA Pro, so you can insert video and data collection into an experiment simultaneously. So that's just to let you know that there are experiments with video that exist in GA Pro themselves. And then before we go to looking at steps, let's just take a look at that. So what I'm going to do is let me disconnect here. And I'll say file, new experiment. Now you see here it says there's sensor data collection, data sharing, and manual entry. I can also choose to see sample experiments. And we have some chemistry ones and then some biology ones. And then I'm going to take a look at control of human respiration. Let's just download that really quick. Part 1 and take a look at it.
And we have a really quick question. Carrie says, I noticed that you did not use a nose clip with either sensor. Is the data similar without them? I have had students with nose jewelry object to nose clips.
I have never seen, like, for best case use, you don't need to use a nose clip with the respiration belt ever. Not required. For spirometry, it's good to use a nose clip just so that you can maximize the air that's moving through the mouth and not the nose. But I have never really seen a difference. I don't like using a nose clip, so I don't use them. To me, they're not critical. It is best use case. We do have nose clips that you could use and put on your nose, and that you can use. And, best use case would be to use a nose clip. I would just sound really weird if I had one on. In my experience, I have some people that use them and some people that don't. To me, it's one half dozen of the other. If people don't want to use them, you can just tell them to make sure they don't breathe through their nose. You still will get decent data, right? These are not clinical. None of the equipment that you're using is for clinical use, and so it should be fine. Yes. So yeah, you can use the nose clip if you want. I often don't. Some people do. It just really depends. Funny, when I go to use a spirometer, actually at my doctor's office, they don't have me use one, but if you want to teach people, like, the best use case is to actually use a nose clip, and you can put one on, and they work fine. All right, so in this case right here, what I wanted to show you is this is my friend Walter, and he's wearing the respiration belt. This is the data that we collected doing the same experiment, so it's a longer experiment. But what's neat here is I can enable replay, and then I can make this experiment happen a lot faster. And then what I'm going to do is I'm going to play. So what we're going to do is we're going to see the video and the force and respiration rate happening in real time. So yeah, this is Walter breathing really fast. Don't worry about that. It's actually, he's breathing slower, but you can see here's the data coming across. Here's the respiration rate. And then there, he held his breath. He's holding his breath. Then he begins breathing again. And there's the data that comes across. So you can create these experiment files yourself. You can stick this data. You can take video, collect data at the same time, stick it together. That's a really useful thing that you can do. I just wanted to let you know that these experiments exist and that you can create them yourself. All right. Other questions on how to use the respiration belt? Now, another really cool thing about the respiration belt is it's also a pedometer. And I just want to show you one of my favorite experiments that I've done that I think is really neat. And we can measure steps and step rate with the built-in pedometer. And so in addition to using it to measure respiration rate, you can also use it to measure the number of steps and step rate. And when I used to be a runner and I ran a lot, I really got into McDougall's book about barefoot running. And so I've done a whole bunch of experiments looking at running shoes and barefoot running, and even comparing different types of shoe types. And what's really interesting is if you run barefoot, you take more steps. Your gait is faster. So you have a faster step rate than with really cushy running shoes. And you can easily duplicate that at the track, out in the parking lot with the Go Direct Respiration Belt. All you have to do is have a runner that can run very consistently. They just have their known gait. What you do is you just run at your standard tempo pace or whatever you want. And then you just have them run barefoot with their running shoes. And then what I would do is get different types of running shoes. So usually I would be the subject because I was at a very standard pace. And then I could run barefoot. I could run with some really low profile shoes. And then I would run with my Oka Ones, which are like really, really have a ton of padding in them to watch how all these things change. What's great about this is this lab, there's a barefoot running lab that exists in the Human Physiology book, volume 2, but it's also free. So we have it freely available on the website. So I'm going to take you to it here. Let's see. Someone said, have you seen a difference in students understanding when they use the automatic calculations versus counting? That's a great question. And I love that question. Me personally, when I taught as a former instructor, I liked using the EKG sensor and a respiration belt that just showed respiration effort. And then I made them calculate everything. Like I wouldn't let them use any auto calculated channels until they proved to me that they knew what a rate was, how to calculate a rate, and that, it's a mean over a period of time. But it's really just up to you. I'm a college professor, and that was like one of my favorite things to do is I would turn around and say, okay, we're going to look at raw data. Now I'm going to show you how we calculate rates. And then once they had, figured out and understood what all of those things were, then I would let them use auto calculations. But I was also described as mean, because I would also make them do a lot of math. So it really just depends on your teaching style. I really liked doing that type of stuff, making them look at the raw data, calculate means, making sure that they understood what they were measuring. But we don't all have time for that either. So anyway, this lab is free. You can investigate the biomechanics of running. It's very similar to the lab that's in the book. It's free. There's a free download of it. And really what you're going to wind up seeing is the experiment basically just has you go out and run around and basically look at the step rate. And how you do that is really simple. I'm not going to go run, but let me do a new experiment. You would put the respiration belt on. You just put it on. I'll turn it back on again. The students connect to it. And then the key thing here is that instead of looking at respiration rate, which is really hard to do if you're running, you're not going to be able to use it as a respiration belt if you're running. But what you can do is turn these channels off and turn on steps and step rate. And then what you'll see is it'll show you the total number of steps, and then it'll calculate the step rate, which is the number of steps per minute. So it'll actually calculate the step rates or the step rate. So if you actually wanted students to calculate step rate automatically, just use steps. If you want them to look at the step rate as it's calculated by the device, use step rate. The only thing to be aware of is you are going to need to zero the steps. Because every now and then as you walk around, it will actually grab a step or two. So before you start the experiment, you can tap here and zero, and that just tells the sensor, hey, I'm about to begin. I want you to take any steps that you've seen and zero them. And you can see that was me shaking it around a lot. So it said, oh, the step rate's 169. It'll go back to zero. Don't worry about that. Oh, why can't you use the respiration belt for running? That's a great question. It's because it bounces around so much. So if you think of it, let me go back to the presentation here. Oh, here we go. If you think of it, when you're running, each step is going to be hitting and pounding. And what winds up happening is the respiration belt, even though it's measuring force in just one direction, that's still enough force to create a lot of noise. So we do not recommend it for running. I think I have gotten it to work for walking, but I really wouldn't recommend it for like full on running, sprinting, or things like that. It's just not going to work very well for that at all. Other questions? All right. So there's no other questions about how these two devices work? Other things? Let's see here. I think it would be fine on stairs. If it would be fine on stairs if you're walking. It probably would be fine on stairs if you're walking. It's really running that would be problematic. But I think if you were walking slowly, it's probably going to be fine. If you want to use it for that purpose and you're just going to be walking slowly, it should be fine for that. You can also just get in touch. It would work great for biking, right? You're not going to have the same issues at all if you're going to wear it while you're biking, and then increasing your exercise and seeing how it changes. Yeah, go ahead and feel free to present some feedback. Dylan's going to be putting that there, and then just let me know if you have any other questions. If there's things you want me to talk about, things that need to show you, or things that you're interested in. One of the really cool things is that if you have any of our older sensors, a respiration belt, or even if you had a force sensor, I can show you how to turn your old force sensor into a respiration belt. It's really not that hard. If you have the old gas pressure sensor and the bladder that can be used as a respiration belt, I can show you how to make that turn into or measure respiration rate as well now using graphical analysis. It has a bunch of great features in it. You would need Graphical Analysis Pro, but I could show you how that would work. All right. Well, if nobody has any other questions, I think that's it. Let me just show you a few other things. I'm going to skip data sharing. I just wanted to let you know that we do have an entire package. Definitely check out Vernier Graphical Analysis Pro. The human physiology package includes EKG sensor, respiration belt, spirometer, oxygen gas sensor, hand dynamometer, a heart rate monitor, blood pressure, and this thing right here is a reflex hammer. It's a reflex hammer and a force sensor. You don't have to buy the entire package. You can buy each individual item a la carte. So don't feel like you have to buy the entire package if what you need is just a few sensors, but if you want the whole package, you could get that too. Let's see, other questions. Oh, great. I'm really glad you've had great results with the EKG sensor. Yeah, the EKG sensor, a lot of people have told me it works great. We've put a lot of effort into it. We try to make sure that all of these sensors will work great for you. The hand dynamometer is another great one. The reflex hammer is another one that people are really excited about. Thank you, Carrie, for saying that the EKG sensor is working great for you. Any other questions? If there are no other questions, I want to just say thank you to everyone that's here and for sticking out the whole time. I really appreciate it. And please get in touch if you have other ideas for webinars. If there's like a webinar that you would like to see or a subject, feel free to email me at biology@vernier.com or put it in the chat, and Dylan and I can talk about it later.
All right.
Hey.
Thanks,
All right,
everybody.
Thanks so much.